Hello ladies and gentlemen, welcome back to C.A.R. Entertainment. I am your host Dylan Hamilton, and today we're going to be doing A21 biology revision, guys. This time we're going to talk about ultrafiltration, reabsorption and osmoregulation. It's going to be very similar to last time when we talked about homeostasis in the kidney. Two different PowerPoints, going to merge them into one. Again, you won't see it through the magic of editing the switch over, but let's get straight into it. Basically, we're just going to finish off the kidney completely here, as ultrafiltration and reabsorption are the two real parts of sort of the excretion function, and then we also have your function in osmoregulation. So let's just get straight onto this. So function 1. So kidney function 1 is filtration, which is producing urine, and the two main functions within this is ultrafiltration and reabsorption. So if it asks you to state what are the functions at this certain point, you'll be using ultrafiltration, reabsorption, and osmoregulation. Okay? So what exactly are these terms? Right. Well, ultrafiltration is the filtration of plasma and other substances below a certain size into the Bowman's capsule. Reabsorption, however, is different, as ultrafiltration is based purely on molecular size. So reabsorption is when these products are selectively reabsorbed back into the bloodstream from the nephron, and it is essential that the useful products that get filtered through are reabsorbed. So we talked about hydrostatic pressure a little bit last time within the kidney, and why there is a hydrostatic pressure, and basically we're now going to talk about why it comes at a high hydrostatic pressure. Essentially, it's really because there's a short distance from the heart that the blood travels down to the kidney. It comes from the aorta, then the renal artery, and then straight to the kidney. So it's quite a short distance. We also know that the afferent arteriole is wider than the efferent arteriole, so whenever blood comes in, there's a lot of it there, and it's at a high pressure, and then it gets there's less of it coming out. Then you also have a coiling of capillaries, which restricts blood flow, which increases pressure as well. Now, the high hydrostatic pressure will force glucose, amino acids, salts, water, and urea into the Bowman's capsule. That's a key point. So ultrafiltration then, it's when we need to talk about larger components. So larger components such as blood cells and plasma proteins are too large to pass through. And ultrafiltration is aided by the structure of the capillary walls of the glomerulus and the lining of the Bowman's capsule, as there are squamous endothelial cells with small pores, and the Bowman's capsule is lined with podocytes. Now, podocytes can extend into two planes, allowing the filtered material to pass through easily. And there's another filter as well, along with these endothelial cells and podocytes, and this is called the basement membrane. And this is an effective filter which determines which components enter the Bowman's capsule more so than the other ones, as the basement membrane will prevent the blood cells and plasma proteins leaving the blood. Now we're going to talk about a little bit about the filtration force. So for filtration to occur, your water potential within the glomerulus capillaries must be higher or exceed the water potential within the Bowman's capsule. And this is because the glomerular filtrate will have a more negative water potential. Okay? So your water potential in the glomerulus must be higher than the water potential within the Bowman's capsule. So basically it must have a more negative water potential because your glomerular filtrate has a more negative water potential. So glomerulus should be maybe minus 250, Bowman's capsule minus 100. Okay, you sort of get that. So this hydrostatic pressure or pressure potential is greater than the hydrostatic pressure, back pressure created by the filtrate. That's a wee bit confusing, so you don't really need to know a lot about it. Now, solute potential is represented by plasma proteins in the glomerular capillaries, but not in the filtrate. And this is because these plasma proteins don't filter through. Now, because of this, your filtrate will have a less negative solute potential, and therefore there'll be a difference in solute potential which opposes filtration. Okay? So net filtration then is a pressure which causes fluid to move from the glomerulus capillaries into the Bowman's capsule. So you talk about water potential and solute potential together, and you'll have a net filtration pressure which moves this fluid from the glomerulus into the Bowman's capsule. And your hydrostatic pressure also will help. Now let's talk about reabsorption. So useful blood products will be reabsorbed as the filtrate passes along the proximal convoluted tubule. Glucose and amino acids will be selectively reabsorbed by facilitated diffusion and active transport. Now, as glucose and amino acids and even some salts are reabsorbed, an osmotic effect will be created, causing 70% of water in the filtrate to re-enter the blood capillaries by osmosis. Small plasma proteins, smaller ones, will make it through, and these will be reabsorbed by pinocytosis. Now, there will be epithelial cells instead of endothelial cells lining the proximal convoluted tubule, and these will have a high metabolic activity which continually carry out active transport. The filtrate will reach the end of the proximal convoluted tubule where no glucose or amino acids will be present as they've all been reabsorbed, and the concentration of urea in the filtrate will increase along the length of the nephron due to the reabsorption of water. This is because if you take water out of something, the concentration is stronger as water is a dilute. It dilutes something. So if you added more water, then it would become less concentrated. Take it out, more concentrated. So the proximal convoluted tubule has different adaptations. It's adapted by having microvilli, which increases surface area for reabsorption. It also has mitochondria, which provide ATP for active transport, and it also has the infolding of a membrane, which further increases the surface area. Let's move on to osmoregulation. So here we are now on osmoregulation. We're just going to get straight into this. Osmoregulation essentially is the controlling of water balance in the body. Now, the collecting duct is where water regulation will take place, second mostly to the proximal convoluted tubule. Most water, as you see here, is reabsorbed in this PCT, but the exact amount of water reabsorbed cannot be controlled. So the reabsorption in the collecting ducts is controlled, and this is done by varying permeability of the collecting duct walls. There's this hormone called the antidiuretic hormone, or ADH, which will control the degree of permeability of the collecting duct walls and also the permeability of the distal convoluted tubule. So, we'll talk about the loop of Henle. This isn't really osmoregulation. This is more reabsorption, but we just have it in this PowerPoint. So, the loop of Henle. This is a wee story that sort of goes on and talks about the loop of Henle, and it hopefully will make you understand it. So, the isotonic filtrate, when it leaves the proximal convoluted tubule, will enter the ascending limb of the loop of Henle, and it will become hypertonic here because the urea is more concentrated because water is being reabsorbed through the proximal convoluted tubule. But it's also more concentrated because the water potential is lowered as sodium and chloride ions are pumped out of the ascending limb. So if you think of it a diagram where you have the descending limb on the right and the ascending limb the descending limb on the left and the ascending on the right. Your ascending limb will have sodium and chloride ions pumped out, and these then move across and enter the descending limb to its left by diffusion, and water will leave. And this will lower the water potential, increasing the concentration of urea, making the filtrate hypertonic. Okay? So at the hairpin then, this filtrate will be at its most concentrated. And then it will come back up and enter the impermeable ascending limb, okay? And then the filtrate will become hypotonic, as no more water can leave, and therefore it can become more dilute. So ADH then, the role of the antidiuretic hormone ADH. ADH is produced in the hypothalamus, which is then secreted into the posterior lobe of the pituitary body where it is stored. These key terms you do need to know. The solute potential will be monitored by osmoreceptors in the hypothalamus. Now, the solute potential of the blood will be whenever it becomes more negative, this will be detected by these osmoreceptors. And then the posterior lobe of the pituitary body will release ADH, causing the walls of your distal convoluted tubule and collecting ducts to become more permeable. Now, as more water is reabsorbed from the collecting ducts, because when they become permeable, water is reabsorbed back in through to the vasa recta and the capillaries, the solute potential will return to normal, and then a smaller volume with more concentrated urine is produced. Should note that if it turns the question on its head and asks you about caffeine or alcohol, it does the opposite, as it believes you are hydrated, and therefore it releases less ADH. And this means the walls of the DCT and the collecting ducts become impermeable or less permeable. And this means that more water leaves the body. So it's not reabsorbed, it leaves the body. And therefore, you'll have a larger volume of a less concentrated urine because there's more water present. But as you're losing more water, dehydration can occur. I've been Don from C.A.R. Entertainment. Hopefully you've enjoyed this video on osmoregulation and ultrafiltration and reabsorption. Hope you've enjoyed it. I've been your boy Don Hamilton from C.A.R. Entertainment, and I'll see you next time.